You all saw the second homework assignments posted, and it's due on Friday. If you have questions, you can email me. We have office hours and stuff also. I try to make it a little challenging, so you have to, read a paper and interpret a little bit. And I think it's pretty reasonable assignment. So if you have any questions about it, let me know. All right. And hopefully you watched the asynchronous lecture. I thought we're doing some pretty cool stuff. We are definitely wrapping up the mostly synthetic chemistry component, and we're going to be moving on to more pharmacology topics here pretty soon. Got a little bit more I want to get through though. All right. So hopefully this has been enjoyable. I don't know. I know it's useful to review your organic a bit, even if you're not going to be an organic chemist. At least you can appreciate that organic chemistry can actually do some pretty cool stuff related to pharmaceuticals. All right. Okay, so the last video, the asynchronous video, was kind of ending with what's it called? Beta-lactam antibiotics. And I'll kind of continue. I have a couple mechanism slides, and then we're going to move on from there. All right, cool. So we're recording. I think we're recording. Yeah, recording. All right. So this was from the asynchronous lecture. The last little bit of that. So I'll just review what I was talking about briefly, and then I have a cartoon summary of it. So how do beta-lactams work? They inhibit an enzyme. A lot of drugs inhibit enzymes or other proteins, obviously, and they inhibit the penicillins and the cephalosporins. Inhibit something called DD peptidase, DD transpeptidase. It's a bacterial enzyme that's involved in the peptidoglycan, and that's involved in amino acid cross-linking, which I'll show in a second. And then what I was kind of ending with was that how can bacteria resist these drugs and maybe prevent them from working? And a way they can do that is using these enzymes called betalactamases, which are enzymes that have a serine, a nucleophilic serine, and they kind of just attach. They attach, or actually no. Let's say that differently. They destroy the beta-lactam. Okay. And they do it in a way that they it cleaves the beta-lactam and then it releases it. Otherwise, this enzyme wouldn't be very useful, right? Because it would say that it got attached and wouldn't let go. Well, then the enzyme would kind of be deactivated, right? Does that make sense? Okay, so. So being a good enzyme, it does its thing. It attacks the beta-lactam, sort of destroys it, prevents it from interacting with this enzyme, and then it releases it because it's just an ester linkage, right? And esters are usually pretty easy to cleave, and that's a pretty effective defense mechanism for bacteria to kind of destroy these drugs that are kind of seen as an enemy of the bacteria, right? So anyway, how can we prevent that? The idea is we can actually use beta-lactamase inhibitors. So these are drugs that are kind of like sacrificial beta-lactams that serine 70 can go attack, but in a way that it seems to be kind of irreversible, right? So serine 70 will attach to a beta-lactam and then kind of not let go. And so now we've kind of destroyed the effect of this betalactamase enzyme, right? That's kind of the idea. And then there's also this thing called sulfactam. It's a similar drug, okay? And also, you know, you can envision that the drugs can kind of combine a beta-lactam antibiotic and a beta-lactamase inhibitor. And a couple of those were clavulanic acid, which is one of these, and amoxicillin, which is a penicillin derivative. That's called augmentin. So it's just like an antibiotic cocktail. And sulfactam and ampicillin is called unison. So it's a pretty cool sort of drug strategy to essentially knock out DD transpeptidase as well as beta-lactamase. It's cool. Yeah, that's a pretty logical strategy. Okay, so we're cool with that. Any questions? And what's the name of this functional group? Actually, where did I see that? Sulfonamide. Oh yeah. Why did I say sulfonamide here? Oh, that's because this is not a sulfonamide. This is a sulfone, right? It's a sulfone functional group. And a sulfone functional group is kind of like a sulfonamide, but a sulfonamide is a little more like an amide, so it's got a nitrogen on it. All right, that's what I kind of got through in the end of the asynchronous lecture from last time. All right, cool. We're cool. Great. Okay, so then I have a cool cartoon diagram that talks about this a little bit more. Let's see if I can find it. Do you guys learn this stuff? I don't know. I mean, I'm sure probably maybe a biology class. It is a little more on the pharmacology side, but I've always been interested in how these kind of drugs work. Okay, so okay, so yeah, this was the diagram of the general general classes of antibiotic drug targets. Oxalinic acid was a gyrase inhibitor. Folate synthesis was targeted by sulfadoxine, and the dihydrofolate reductase was targeted by trimethoprim. And then the beta-lactams, penicillins, and cephalosporins are basically preventing the synthesis of the peptidoglycan, okay, which is in the gram-positive membrane. Gram-positive bacteria. It's the outer cell wall. Now, in the gram-negative bacteria, it's a little more of an inner. It's a inside barrier into the bacteria. Okay. All right. So this cross-linking thing and how does this work, and how do the beta-lactams basically inhibit the synthesis of the peptidoglycan? All right. Okay, so the peptidoglycan is composed of alternating amino sugars. So it's a sugar with an amine on it, and it's cross-linked by this enzyme, DD transpeptidase. Okay. So there's two sugars. One is called N-acetyl-muramic acid, or NAM. The other is N-acetylglucosamine, NAG. Okay, and these kind of alternate, right? So you have this sort of alternating subunit with two amino sugars, because normally sugars are not amines; they are just have oxygens on them, right? And on the NAM, this actually has a peptide kind of linked onto it, and it's, you know, a couple of amino acids, alanine, glutamic acid, and a couple other amino acids. Yeah, the other one does not. So there's not a peptide on the NAG thing. So it kind of alternates back and back and forth. Okay, and the tetrapeptide is usually L-alanine, D-alanine, right? That's kind of weird. D-glutamic acid, D-glutamine, L-lysine, a couple other amino acids are used. So that's a little weird that a bacteria would use a non-natural amino acid in its peptidoglycan. Any guesses why that might be? Why do bacteria use non-natural, like D amino acids, when they build these kind of shields around them? Any guesses? I mean, do humans regularly use D amino acids? We're kind of based on L amino acids, right? You guys heard that story before? I don't know. Why would D-ala be used as opposed to L-ala? Any guesses? So it's interesting, but bacteria are very defensive little creatures. And they are it's kind of they're evolving, right? And they're evolving to avoid basically, drugs and antibiotics and things like that, and also just host defense mechanisms like proteases that might break proteins down and cleave amino acids. And by using D amino acids, bacteria can try to evade natural defense mechanisms and enzymes and things like that. So it seems to be like basically like the bacteria are evolving, to switch the stereochemistry and maybe prevent destruction by things like proteases, right? Okay. Anyway, so what happens with DD transpeptidase? This enzyme. What it basically does, you can kind of see that we alternate between NAG and NAM, and there's a if you look really closely, the NAG are slightly darker, so that's what we're trying to show. Is it goes NAG NAM NAG NAM NAG NAM, right? And anyway, the NAM units have these 4 amino acids, and they can be kind of cross-linked. So essentially, these chains are being built, right? And then the cross-links will create like peptide linkages and other linkages between the 4 amino acids of one chain and the 4 amino acids of the other. So it basically builds a really rigid wall, right? So it's like it's like a something that can be not permeated through. So that's basically it. So it's a cell wall, right? It's a type of cell wall, and essentially that's what we're showing as this kind of purple thing in the gram-positive bacteria. Gram-negative bacteria also have it, but it's a little smaller. And the gram-negative also have an outer cell wall. Okay. And then the beta-lactams are essentially inhibiting this enzyme, and that's how they work. And that's how, if you take an antibiotic like if you have an infection or something, that's essentially what they're doing is preventing the cross-linking of the cell wall, and that is lethal to the antibiotic, to the bacteria. Okay, is that it for this? That's it for this. All right, so we're done with that. Cool. All right. Give me 1 second. All right. So just a tiny bit more on these beta-lactam antibiotics. So we're still on number 6, which based on our organization is related to the synthesis of these drugs, carbonyls, enols, aldols. Okay. So let's just say one more thing about these drugs like clavulanic acid. Sulbactam, etc. So they were the beta-lactamase inhibitors. All right, and so as a class of drugs, these are kind of weird, right? Because what are they doing to, what are they doing? They were permanently attaching to an enzyme, right? Beta-lactamase, serine 70, right? Does anybody know what that's called? Like the sort of the covalent inactivation of an enzyme? Is that common when we think of drugs? Like, do drugs like irreversibly attach to enzymes? They usually kind of attach like non-covalently and maybe reversibly inhibit the enzyme. But what's it called? Anybody know where a drug permanently attaches to something? Have you ever seen that before? So if you haven't heard of it before, it's kind of a cool idea, right? These are called suicide substrates. It's the classic name for them, and the idea is they are as molecules, they're destroying themselves by getting basically permanently attached to the enzyme, and that's a unique mode of action, right? So let's see what do we mean by that. So let's show our beta-lactamase again. Beta-lactamase. Here's serine 70. Hydroxy group. Clavulanic acid. There's a beta-lactam. Carboxylic acid, primary alcohol. And like I said, what actually happens is right out of your organic chemistry textbook, the alcohol attacks, and then, electrons kind of go onto the oxygen, go back, break open the beta-lactam. That's what we love to call NAS. I certainly love to call it that. Not everybody does. NAS, nucleophilic acyl substitution. Oxygen attacks, electrons onto the oxygen, they break back off and cleave the beta-lactam. And then you've kind of attached to serine 70, right? Then you have CH2, oxygen, NH now, CO2H. There's your carboxylic acid, CO2H, yeah? CO2H, and then the primary alcohol. Okay, cool. Yeah, it's like it's an alkene, CH2OH. All right, cool. And it does not let go. That's right. Does not let go. I mean, the funny thing is, if you look at the other beta-lactams, like amoxicillin or the cephalosporins and all that, they also have a beta-lactam. So what makes these special? Like, in that it does not let go? It's a great question, right? Because if beta-lactamases function in their existence is to destroy beta-lactams and then let them go and keep doing that, what makes these special? It's something about the structure of them. I don't really have a good answer for that. Maybe somebody has studied that using a crystal structure or something. But essentially, this thing's a stable entity. This ester is stable, right? Stable ester. Weird, huh? That this ester is stable, but the other ones with penicillins and cephalosporins somehow are unstable. So there must be something about the rest of this that kind of stabilizes it, or within the active site, it's stable. Okay, so what about sulbactam? It's a similar idea. Sulbactam, which is another one of them. Beta-lactam. We got the sulfone up top. 2 methyls, carboxylic acid. Yeah, so it does the same thing. It attacks, it's an NAS type thing, and then serine 70 has that thing attached. Nitrogen. Now even the nitrogen from the beta-lactam is now here. And then we have the carboxylic acid and the 2 methyls. Okay, cool. Anyway, do you expect this to be a common type of drug? And what might be wrong? What might be bad with a suicide substrate drug? Any guesses? I mean, if you're permanently modifying enzymes, that could be bad. I mean, there might be a toxic side effect or something like that, right? In this case, you're deactivating an enzyme from a bacteria, so it's probably considered good. You're trying to mess up a bacteria, so this is a reasonable approach for antibiotics. But anyway, there are more examples of suicide substrates, and if you just want a little taste of this, you can do Wikipedia search is actually pretty reasonable for suicide substrates. We will mention other ones in this class, as we, you know. I think omeprazole. I remember omeprazole. Remember that, the proton pump inhibitor. I think it's actually formally a suicide substrate because I think it does actually attach to the enzyme covalently. But there's a series of them that are all used, you know, in clinical applications. Okay, cool. All right, are we done with that now? I think we might be done with beta-lactams. Okay, we're done with beta-lactams. Any more questions on these? All right, so we basically have one more synthesis discussion, and then we're on to pharmacology. But I'm going to do a quick tangent into a little bit more pharmacology, and this may help you on the homework a bit. So we're going to do a little tangent. One more example of some reactions, and then it'll be pure pharmacology. All right, cool. So I'm calling this tangent number 2. Because we did a tangent number 1 earlier, which was on peptide coupling. This is what I'm going to call brief tangent number 2. So this is going to be on very basic pharmacology concepts. Definitely helpful for your homework. I just want to basically discuss a little bit more on the concepts of IC50, EC50, ED50, TD50, LD50, and LDLO. Okay, what are these different things? Let's just define them real quick. Definitions. I mean, we've seen a couple of these already, so. Definitions. So IC50, of course, is the drug concentration. So, and what is concentration? It's like nanomolar, micromolar, etc. So it's NM, UM, etc. So it's a concentration, right? Units. Drug concentration producing 50% inhibition. And so you, like for most drugs, we want it to be a low value. So, you know, low nanomolar, maybe low micromolar or something like that to be potent, right? Okay. So how does that differ from EC50? And what does EC50 mean? Anybody know what EC50 is? It's something about 50% something, right? Anybody ever hear that before? EC50. And how might it be different than IC50? So EC50 is a drug concentration as well, producing 50% effectiveness. Well, how is it different? I mean, if like you would think that maybe inhibiting something 50% would basically cause a 50% effectiveness. Why are these different? Why don't we just use one of these? Any guesses? What might be unique about an EC50? Or what kind of drug would you use and describe potency with EC50 versus maybe a drug target where you use IC50? Have you thought about this before? Maybe you haven't seen this before in a course, so it's good you're learning it, right? Nobody knows? So it basically comes down to this. Not every drug target our goal would be to inhibit it, right? So sometimes you have an enzyme you want to inhibit, right? Sometimes, or a channel protein or something, it's like you want to inhibit the function of something, right? But what if you're trying to what if your goal is to activate it, right? It's the opposite of inhibition. You want to activate an enzyme or activate a channel protein or something like that. In that case, IC50 doesn't make sense, right? Because this is specifically for inhibition. So EC50 is the measure of potency you would use. Okay. All right. ED50, of course, we talked about D versus C. What's D? What's the difference between C and D here? ED50. Just dosage, yeah. Yeah, dose, yeah. So It's the drug dose. It's the drug dose producing 50% effectiveness. Okay, yeah, cool. And then of course, we've talked about some of the maybe undesired effects of drugs. Obviously, TD50 is is basically a measure of a dose that would produce toxicity among 50% of a population. So this would be a drug dose producing toxic effect, some kind of toxic effect. It could be cardiovascular toxicity. They could produce some kind of neurotoxicity or something. So it's a little bit generic. A toxic effect across 50% of a population. Now that could be, I mean, frequently they would use something like a rat or a mouse. It could also be used, it could describe for a wide scale study an effect in humans as well. Usually we'll say rat or mouse. Okay, cool. And as we mentioned before, LD50 is a little bit beyond toxicity. It would be a lethal dose, drug dose producing lethality across 50% of a population. Okay. And then we have LDLO. This is another interesting one in terms of drugs, and this is a very commonly known for a lot of drugs. It's the lowest dose to cause lethality. Lowest dose to cause lethality. So this is known for a lot of the common drugs, like clinical drugs. Of course, it can be measured for a rat or a mouse, but also definitely human, because a lot of drugs that are commonly used do have a known lethal dose, even in humans. And the lowest dose would be the lowest, a dose that if you like the lowest possible dose that might actually kill a human. And so obviously you're hoping that the effective dose is much less than that, right? So and then it's generally considered safe. But like drugs like acetaminophen, Tylenol, have actually a measurable lethal dose in humans, and we try to avoid that. And those drugs are also kind of toxic, and you can easily overdose and basically either have permanent liver damage or basically die from it. And so having a nice separation between an effective dose versus a lethal dose or a lowest lethal dose is desired. Okay. All right, so let's keep going. And we talked about this before. Let's actually show how IC50 relates to drug concentration and inhibition. I think we plotted this curve before. Let's do it again. Okay, so IC50. The easiest way to visualize this is a curve with some kind of effectiveness or of the enzyme or protein you're inhibiting. we'll say percent activity of like an enzyme, 100% active, 0% active, right? Okay, and you can imagine that if you have a very low dose, like 1 nanomolar of a drug, that there's no effect at all. And then you do have 10 nanomolar, you might have a little bit of an effect. And 100 nanomolar, maybe more of an effect. 1,000 nanomolar, which would be 1 micromolar, more of an effect. 10,000 nanomolar, maybe complete inhibition, right? So you have this kind of curve, and then you can kind of plot it. And the idea is that the 50% point defines what the IC50 value is. So this kind of drug, generic drug example, the IC50 would be about 100 nanomolar, right? Okay, and so typically you would want that to be a low value, and you don't want it to be a large separation between IC50 and like lethal or toxic concentration or dose. Okay, all right, so lower is better. Lower is better. More potent. Cool. As an example, 5-fluorouracil is a anti-cancer drug. Let's call it 5-FU for 5-fluorouracil. It's, as an example, it's potent against all sorts of cancer cell lines. It's also used clinically as a chemotherapeutic drug. As an example of IC50, it inhibits something called SW620 colorectal cells, colorectal cancer cells, cell growth. And it has a specific IC50 value. So the IC50 of 5-fluorouracil is about 500 nanomolar. NM, right? Nanomolar. What does NM mean? What's it like as a concentration units? What does that mean? It means it's 500 nanomoles per liter, right? Nanomoles per liter, or you could say 500 nanomoles per 1,000 milliliters, which of course is the same thing. Okay, so that's the meaning of nanomolar. You guys should all know that from Gen Chem. Okay, what's the structure of 5-fluorouracil? It's a simple molecule, and this is definitely used clinically to treat a lot of cancer types. It is toxic. It's very toxic, but it's very effective also. So it is it's a uracil derivative with a fluorine in it, and it basically gets incorporated into growing DNA. And of course, in cancer, DNA is growing like crazy. And when this gets incorporated into growing DNA, it kind of terminates it because the fluorine is a non-natural atom, and it basically terminates DNA polymerization. Okay, this is super toxic, and so there's a lot of less toxic cancer drugs nowadays. But this would be a drug that would definitely like cause hair loss and things like that, and as a chemotherapeutic treatment. Okay, very, very toxic. All right, good. Let's see. Okay, so let's show EC50 now. An example of EC50. So EC50 again is something we might use to describe like rather than inhibition, maybe activation or agonism. Okay. Instead of an antagonism, we can describe agonism. So similar to IC50, but for effectiveness generally. And used to quantify potency for agonists. Usually, if you're describing inhibition or antagonism, you would always use IC50. But in the case where you're describing an agonist, it's usually going to be EC50. All right. So of course, let's just look at it as a general example. The percent response, we say percent response on the y-axis. So 100% response, 0% response. Okay, so 1 nanomolar, 10 nanomolar, 100 nanomolar, 1,000 nanomolar, 10,000. Okay, so in this example, we'll just say, okay, low concentration, no effect. And then as we kind of increase the dose, then it sort of plateaus off and we get a maximum response. And then in this case, the 50% mark will describe the EC50 value, which in this case, arbitrarily, we're saying, it's about 10 nanomolar, right? EC50 equals 10 nanomolar. Okay, so that's pretty easy to understand. As a real example of this, I'll actually mention a drug called rizatriptan or Maxalt. It's actually a drug that I use as an anti-migraine drug. I don't know if anybody else suffers the fun of migraine headaches. Migraine drug. This is one of the pretty much very common modern anti-migraine drugs. It's actually quite effective, and it is an agonist. It's not an antagonist. Okay, so the structure of it's kind of cool. Benzene. It's an indole. Indole with a nitrogen and 2 methyls. These are called tryptamines. This molecule is called tryptamine. So, and this is related to tryptophan, right? Tryptophan, which is the amino acid, where you have like a carboxylic acid right there, the NH2. But this is a dimethyl tryptamine, and it has a nitrogen. Anybody know what? Remember what heterocycles? This heterocycle is called. It's a 3 nitrogens, right? What's that called? Well, if there's 4 nitrogens, it's called tetrazole, right? 4 nitrogens is tetrazole. 3 nitrogens is, any guess? 3 nitrogens?
Triazole?
Triazole, that's right, yeah. Imagine if you had an extra nitrogen, then it'd be called tetrazole. If you have 2 nitrogens like this, it's called imidazole, okay? But anyway, that's called triazole, yeah. Anyway, it's a pretty simple drug. Pretty simple drug. It is a agonist, meaning it activates the serotonin receptor, 5-HT, 1B and 1D. 5-HT is serotonin, 5-hydroxytryptamine, I think it is, 5-hydroxytryptamine. And It's one of the neurotransmitters. There's a bunch of subtypes of 5-HT. So there's 5-HT1B and 5-HT1D. And somehow these subtypes of the serotonin receptor, 1B and 1D, effectively, if you activate these serotonin receptor subtypes, it seems to have a pretty effective treatment against migraines. How that actually works, I mean, there's probably there's a ton of biology behind migraine headaches, and somehow people figured out that these specific receptors are if you activate them, they kind of prevent the development and pain of a migraine, which is pretty cool. Yeah, okay, so and so these are the target of a bunch of different triptans. So there's different triptans that have the general structure of this target of tryptan migraine drugs. And being something you activate as opposed to inhibit, this would be a case where there's an EC50 value, and the EC50 is a known value, and the EC50 is 90 nanomolar in humans. Also, if you get prescription pharmaceuticals, they often have a little booklet inside, and they actually will mention things like EC50 values. It's pretty well, it's pretty common to see that in modern pharmaceutical, like the little pamphlets and stuff that you get. Okay, so let's go next to ED50, which is the effective dose. And we'll continue this example of rizatriptan. Okay, so back to rizatriptan. And ED50 again is the dose of it. So if we consider rizatriptan, the ED50 is 54 micrograms per kilogram of body weight. Okay, that's actually pretty small, right? We think, micrograms are very, very, it's hard to measure a microgram, right? Right? It's what is it? It's a thousandth of a milligram. So how do you measure a thousandth of a milligram? It's kind of difficult. But I mean, the thing is, where the actual dose will be larger because a typical human is not a single kilogram. So what's the weight of an average human? About 90 kilograms or so, plus or minus. A lot of variability, but per average human. Okay, so we do the math. It's at about 4860 micrograms per human. And actually, it's which is how many milligrams is that? That's 4.8, right? So what's the normal dose of rizatriptan? It's actually 5 milligrams. So like me as a migraine sufferer, the actual dose I take is a 5 milligram dose. So it's pretty close to this 4.8. As just another, this has nothing to do with really chemistry, but just a little fun fact. The cost of this drug, it's a pretty simple drug, is about $500 for a box, which is about $50 a dose, which is pretty ridiculous for a really, really simple drug like this. But it just shows how much markup that the markup of drugs, it's a very simple drug like this that could be made in like 2 steps from simple reagents. But you know, with insurance and everything, it's about $5 per box. So, but people that don't have insurance are paying a lot for a simple drug like this. Okay. All right. So that's ED50. Let's talk a little bit more about LD50, TD50, and the LD low, the lowest observable lethal dose. Okay. LD50, TD50, LD low. Okay, so let's just mention, we already talked about what these mean. It's the lethal dose or toxic dose for 50% of a population. You could say these are measurement of toxicity. I mean, overall, you want a drug to be, you know, you don't want the effective dose to be anywhere near the lethal dose or toxic dose. We'll say roughly measurement of toxicity. And an interesting case example for these is acetaminophen, which is Tylenol, right? And Tylenol is definitely a drug that can be you could suffer an accidental overdose by taking too much of it. The toxicology of it's pretty interesting, and it's all related to the chemical structure. It's a very simple molecule. Tylenol is this. It's just a hydroxy group para to acetamide. The toxicity, and I think we're going to talk about this a little bit later, is all related to the fact that this can be oxidized to a benzoquinone, which is something like this. And this has extreme liver toxicity. And liver enzymes can like microaddition to this and things like that. That's kind of one of the methods of toxicity. And we will talk about basic toxicology of some substances. But this kind of arrangement, whenever you have two heteroatoms para to each other on a benzene, you have this potential for oxidation to things like that look like this. And these are really toxic and liver toxic. Okay. All right. So what is the LD50 of this? This is a very well characterized drug because it's been used for, I don't know, 50 years, maybe probably longer than that. Maybe 60 years as a NSAID drug. NSAID drug. Not all NSAID drugs have this kind of liver toxicity. Like ibuprofen doesn't. Ibuprofen has other like kind of stomach toxicity. But this is definitely a liver toxin. High concentrations can lead to liver failure. Okay, so what's the LD50 of it? LD50 in a rat is about 1944 milligrams per kilogram oral dose, because it'll taking a drug intravenous would usually taking a drug orally will prevent absorption of some of it. So taking a drug intravenously, you would actually get a higher dose, and probably the lethal dose would be a lower concentration, or a lower dose actually, because as drugs are taken orally, a lot of the drug kind of never gets absorbed into the bloodstream. Okay. Okay, so then so for example, LD50 rat, intraperitoneal is another injection route. It's the peritoneum is like a cavity that you can sort of inject drugs into, usually in rats and mice. It's not into the vein, but it's kind of like it's a space in the body of a rat, and it's a convenient kind of location to inject substances. Anyway, it allows you to kind of avoid the sort of oral absorption and all the complexities of oral absorption. So you can basically inject the substance directly into the body, and then it will be absorbed into the bloodstream. And as you might expect, the lethal dose will be lower because it's more efficient, right? But this, when we develop drugs for humans, we try to actually make orally available drugs. And but anyway, this intraperitoneal is another type of absorption, and of course the lethal dose will be, or toxic dose will be, always lower. Okay, let's just compare this to LD low, which is the in a human. So these are well characterized numbers. This is the lethal dose, the kind of lowest observed lethal dose in a human, and that's about 143 mg per kg. Okay, so that means, like, I mean, across large populations of studies, 143 milligrams mg per kilogram. This would be like the lowest observed dose, and it makes sense that this number would be a lot smaller. In this, obviously rats and humans are different, but this number is a lot smaller because it's not dealing with like a 50% population. It's like just the lowest observed dose, right? And if you assume a 90 kilogram human, roughly, average weight of a human, what is that actual dose? The actual dose, and this is oral also, because typically Tylenol is taken orally, so it's about 12.8 grams. Okay, and there's also a lot of like toxic side effects that are not necessarily lethal for humans, but this kind of these kind of numbers would set the guidelines for like what doses not to exceed to avoid either accidental lethal overdose or a toxic response. I want to say it's about 6 grams is sort of like the absolute. Well, the 6 grams in a human in a day is about that would usually cause severe liver toxicity. Okay, let's see what else about this. As I said, usually, I mean, LD low would be much less than LD 50. That makes sense, because the lowest observed dose would be like for like a single human, whereas 50 would be a, assuming like a larger population. And usually we don't, determine an LD 50 for humans. That doesn't seem to be something that people don't do that, I guess. Okay, let's see what else about this. Just a couple other miscellaneous examples of substances we are familiar with. Sodium chloride, NaCl. What's the LD 50 of that? Is it going to be a larger number or a smaller number than Tylenol? And that might be a good measure of, like, how toxic is Tylenol really, right? So Tylenol is, sodium chloride is about 3 grams per kilogram, LD 50 in a rat. And if we consider sodium chloride as not being very toxic, right? We usually don't think of sodium chloride as a very toxic substance. I guess you could say, you could compare, this is LD50, I'm sorry. Fix that, sorry about that. LD50 equals 3 grams per kilogram in rat. Okay, so you could probably say, well, you know, sodium chloride is not very toxic, and then the LD50 of Tylenol is, you know, it's not that far off from 3 grams per kilogram. You could say that, you know, this is relatively speaking not super toxic actually, and that if we consider sodium chloride not a kind of non-toxic substance. What other drugs or substances are very non-toxic? Anything, you know, sodium chloride does have toxicity because of its effect on like the heart and things like that. But let's say sugar, or I think glucose in particular. LD50 is about 30 grams per kilogram in a rat. And, you know, you say, well, the sodium chloride is definitely a lot more toxic. I mean, or sugar is actually about 10 times less toxic. And then just last example for comparison, ethanol is about 7 kilograms. D50 is about 7 grams per kilogram. Okay, so, and that's also in a rat. So I guess if these compounds, sugars, you know, maybe a good baseline material for a fairly non-toxic substance, right? Cool. All right. So let me see. All right, I'm basically done with this little tangent. I am ready to go into the last drug synthesis example, which is about Lipitor, which is a cholesterol lowering medication. It's a pretty cool topic, but I think it'd make sense if I just stop now and just do that in the asynchronous video. So I think I'm just going to stop now. And I'll present the other stuff as one discrete video. And then we're going to continue on to pharmacology and some of the different factors related to absorption of drugs, different administration routes, pharmacokinetics, pharmacodynamics, and all those kind of things. That'll probably be in the next synchronous lecture. All right. Any other questions about these kind of parameters, EC50, IC50, and those things?
I have a question outside of the topics. It's regarding exams.
Yeah.
So
I think we're going to skip the exam and we're just going to do homework. I think I'm probably going to do that for the course. There was it's supposed to be a midterm coming up slowly, and we're just going to cancel that and just give you a series of homework assignments.
Oh, okay, thank you so much.
Yeah, I think that's the easiest. I mean, all the homework will be in variable difficulty, and you know, so you might consider some almost like a take-home exam, but they're all kind of related to the literature, and you just have to think and read the literature and kind of figure stuff out. But an actual exam, we're not going to do that.
Would it be okay if I can visit you for office hours this week?
Yeah, absolutely.
Okay.
This is Felicia.
Yeah, it's Felicia.
Are you able to come to the normal ones?
Yeah, I can. I can come. I just want to make sure that if you're available on that time.
Absolutely. Yeah, always. Yeah.
Okay, thank you.
Okay. Any other questions?
Yeah, Professor. With regards to those to the parameters those parameters are just for a specific, like for Tylenol, and it will change as like let's see, it will change if we change it for sodium or glucose. So those things will change, right?
Yes.
Oh, okay.
What was the first part of the question again?
Oh yeah. Oh, no, I was just curious because I thought maybe it was just the Tylenol will have like a setup of sort of a number instead of changing. But when you pick, I guess you did an example of the glucose and sodium. I kind of know. I kind of see the difference now.
Okay. Yeah, and yeah, I mean, there's a lot of variability and species variability. There's also like even like the sex of the mouse or rat, things like that. And I mean, there are kind of ballpark, and there's some variability. And I mean, so I'd say it's general guidelines, obviously. Something like the LD-LO that we're talking about, that's a pretty specific number. It's like the lowest observed dose. I mean, that's like pretty specific. But things like the LD50, TD50, and stuff like that. I mean, it can vary a little bit based on other parameters too. So yeah.
All right, got it. Thanks.
Right. All right. Cool. Well, nice seeing you all. And I will get the other video started, and then we're definitely gonna transition here pretty quickly out of the pure synthetic chemistry type stuff, and then a little more into pharmacology. And yeah, we got a lot of other cool stuff to get to. So we're gonna get there. At some point, it's gonna be a little more PowerPoint-ish. I have a lot of PowerPoint for this course, but we're gonna still do this sort of pseudo whiteboard thing for a bit more. And then eventually it's gonna be a little more PowerPoint. And I give you all the slides and everything, all right? Okay, all right, have a great day. See you guys.